So welcome. In this session, we will be looking at iron and minutes. By the end of this session, you should know how iron is absorbed, stored, and transported within the body, recycled, how it is taken up by the cells which require it. How can you diagnose deficiency of iron in the body, and what happens if one has iron overload? Welcome all, my name is Wallace Munyeri and I'm the RECEPTOR. Now, the adult body typically contains about 3.5 to 4.5 grams of iron. 3.5 is females have a lower amount of iron and males have a high amount of iron. Now, iron is a component of many proteins, both catalytic and non-catalytic proteins. Now, the catalytic proteins are like enzymes. Some of the enzymes which contain iron include propyl hydroxylase. Okay, iron is also linked to sulfur to form iron-sulfur proteins, which are found in the electron transport chain. Okay, also it can be part of heme prosthetic group in proteins such as hemoglobin, with in fact 70% of all iron in the body is found in hemoglobin. Also, it can be found in myoglobin. Myoglobin is in the muscles, the skeletal muscles. Also in the cytochromes. Okay. Now, note this: free ionic iron is toxic. Why? It has the ability to produce hydroxyl radicals, and hydroxyl radical is a reactive oxygen species. And remember, reactive oxygen species can lead to they usually oxidize, they carry out peroxidation of the polyunsaturated fatty acids on the plasma membrane, okay, which can lead to cell damage. In fact, reactive oxygen species have been stated to are one of the causative agents of aging, okay. Now, dietary iron is available in two forms. Now, let me take a pen. Two forms can be either in heme or non-heme. Heme makes up just a small percentage of the diet, 10 to 15%. It is mainly from animal sources. But heme iron, although less abundant, it is better absorbed. Note that. Now, non-heme iron, which is in this form, the ferric form, this is the ferrous form, is found mainly from plant sources. Okay. Now, as I have said, the heme iron is mainly from meat, poultry, and this fish. Okay. And the non-heme from plants, such as plant sources, such as cereals, lentils, grasses. Okay. Lentils are these legumes, which we commonly call kamande. Okay. Now, about 10% of the ingested iron is the one which is absorbed. Okay, not all of the one ingested is absorbed. Only 10% of it is absorbed, which should be about 1 to 2 milligrams per day. That is your daily requirement for iron. And the 1 to 2 milligrams is sufficient to replace the iron which is lost from the body, primarily by the sloughing off epithelial cells or the desquamation of the epithelial cells. Okay, as these epithelial cells are being desquamated from the body, they usually carry iron with them, so that is how iron is lost. Okay, and but this 1 to 2 milligram from the diet is sufficient to replace that the iron lost. Now, absorption, storage, and transport of iron. Now, the intestinal uptake of heme, mainly from the animal sources, is mediated by the heme carrier protein. The heme carrier protein is as the name says, is a transmembrane protein found in the plasma membrane of enterocytes in your, especially in your duodenum and the upper part of jejunum, okay? Then within those enterocytes, once this heme is taken up, it is acted upon by this enzyme known as heme oxygenase to release the ion from the heme, okay? Then non-heme ion is taken up by an apical membrane protein known as the divalent metal ion transporter 1, okay? And before we take up this, let me take, before we take it, because it is in the ferric state, we first have to reduce it to the ferrous state. And this is done by this enzyme known as the duodenocytochrome B. Okay, this one. And that cytochrome B is a ferric reductase. You can see it is reducing ferric ion to ferrous. Then it is this, you can see it is a divalent. This is the one which is now absorbed by the enterocyte via the divalent metal ion transporter 1. Okay. Then after absorbing, what next? Okay. It has, after absorption of this ion, either from a heme or non-heme sources, it has two possible fates. One, it can be oxidized to ferric state, and then it is stored in an intracellular protein known as ferritin. Okay, and one ferritin can bind up to 4,500 ferric ions. Okay. Two, or another fate, is that it can be transported out of the enterocyte by the basolateral membrane protein known as ferroportin. Okay, and since it is in this state, the ferrous state, it first has to be oxidized. It has to be oxidized by a copper-containing membrane protein, which is known as hepastine. Now, hepastine, it is closely related to the ferroportin, such that once this ferroportin transports the ferrous ion out of the enterocyte, it is immediately oxidized to ferric state by hepastine. Okay, then what is the function? Why oxidize it to the ferric state? So that it can be taken up by the plasma transport protein transferrin. And transferrin, one transferrin molecule can carry only two ion in the ferric state. Okay, now note this. The other cells, other than enterocytes, they usually contain also a copper containing plasma protein known as celluloplasmin. Okay, they don't have hepastine. So if they want to transport this ion out of their cytosol, say the macrophages, because the macrophages they usually degrade heme also, and if they want to transport that ion in this state, they will first use the ferroportin, but they don't have hepastine like enterocytes. They have celluloplasmin. Okay. Now, in normal individuals, transferrin is about 1/3 saturated with iron. So only a third is saturated. That is about is it 33%? Only 33% is saturated with the iron. The other 2/3 exist in form of apotransferrin. Apotransferrin refers to transferrin to which ion isn't bound to. So 67% is in form of apotransferrin. Okay, let us now see a diagram here. Let me take it back. You can see this is heme from animal sources. It is taken up by the heme carrier protein. Okay, once in the cytosol, it is acted upon by the heme oxygenase to release the ion in it. This ion has two states. It has two fates. Fate number 1, converted to a ferric state and stored in the intracellular protein, ferritin. Okay, let's look at the other one. Now, this ion from non-heme sources, it is in this state, the ferric state. It has to be, first, it has to be converted to the ferrous state by the ferroreductase, the duodenocytochrome B, which is a ferroreductase, and the vitamin C is the coenzyme of this enzyme. Okay, then it is transported into the cytosol of the enterocyte by the diverent metal transporter, diverent metal ion transporter 1. Okay, then it also has the same fate. Now, here you can see it can be transported by ferroportin, okay, out of the enterocyte into the plasma, but it has to be oxidized from ferrous state to ferric state by the hypochromic. Now, this is for enterocytes. In other cells, you find instead of hypochromic, they will have ceruloplasmin. Then you can see each transferrin can bound to ferric ions. Okay. Then you will see that ferroportin is regulated. Its activity is regulated by this hormone produced in the liver known as hepcidin. That is what we are going to look at next. Okay. Here we are. Now, regulation of ion uptake to the plasma. Now, ferroportin is the only known exporter of ion from cells. Okay. And it is regulated by a hepatic peptide hormone known as hepcidin, which has 25 amino acids. And how does it regulate? It usually induce internalization and lysosomal degradation of ferroportin. Okay. Now, therefore, hepcidin is the central molecule in ion homeostasis. Okay. How does hepcidin act? Now, hepcidin in high concentration, it usually suppress. It usually causes the when this hepcidin is high, it means that you have a high ion concentration in the blood. So it will cause the ferroportin to be internalized and be degraded so that the amount of ion taken up to the plasma is reduced. Then when there is ion deficiency, the amount of hepcidin is low. And this will cause more ion to be taken up into the plasma, okay? And so if you take up more ion from the enterocytes, they will also take up more ion from the intestinal lumen, okay? I hope that is clear. Sorry, what am I? The next page, next. Now, recycling. Macrophages usually phagocytose old and damaged red blood cells. You know, after 120 days, the red blood cells are destroyed mainly in the liver by the macrophages in the liver known as the Kupffer cells, also by the macrophages found in the splenic cords of Billard, that is in the spleen. Now, once they are damaged, these usually free the heme ion that is usually sent out of the cells. They also have a ferroportin, and as I have said, they don't have hyperstane, they have ceruloplasmin. And it will oxidize this heme ion, the ferrous form, to the ferric state, so as it is bound to transferrin, and then it will be transported into to the ion requiring cells. Okay, this recycled ion usually meet up to 90% of our daily need. Okay, which is ion is mainly used in erythropoiesis, production of your red blood cells. Here you can see this is a macrophage. It will phagocytose these old red blood cells to free up this ion. Then we have the heme oxygenase here. You can see we require oxygen, NADPH, okay, to produce now this ion, which will be transported out of the macrophage via a ferroportin here. But as it is being transported out, it has to be oxidized to the ferric state. But here you will have ceruloplasmin instead of hyperstane. You can see also here we are producing carbon monoxide, but here carbon monoxide, in fact, it acts as a signaling molecule. It is not toxic here. Then remember heme heme refers to a protoporphyrin, IX, and an ion, the ion at the center. The protoporphyrin IX is usually opened up. See, it was cyclic, then here it is opened up to form the linear molecule biliverdin. Then biliverdin is reduced to bilirubin by the biliverdin reductase. Bilirubin is conjugated to, it is transported out of the macrophage into the blood, bound to albumin. It will go to the liver, where it will be conjugated with the glucuronic acid and excreted via bile. Now, uptake, how is iron taken up by the cells which require it? Okay, now transferrin with the bound ion from enterocytes and also the macrophage usually bind to receptors found on the erythroblast and other ion requiring cells, and it is taken up by receptor mediated endocytosis. Okay, now then this ion is released from the transferrin for use, okay, or it can be stored in ferritin in these cells. Then the receptor, the transferrin receptor, is then recycled in a process which is similar to the receptor mediated endocytosis, as in the low density lipoprotein particles. Here you can see this is transferrin, diferric transferrin, okay. You can see these two dots here are the ferric ions, okay. Then it will bind to this transferrin receptor, this one here. This is a transferrin receptor to form this, okay. Then this will cause like invagination of the plasma membrane at that point, and it is coated with clathrin. Okay, remember this is the one which stabilizes that endosome to mediate receptor mediated endocytosis. Then to the endosome, we will have a proton pump. Hydrogen ions are added to it. Then ion which was bound here is transported out of that endosome. Then this endosome will be somehow like it will undergo exocytosis, so that the transferrin receptor can be recycled. And then you can see here the transferrin does not have it is not bound to ion, hence form apotransferrin. Ion here can go to like can go to form like heme, also can be stored into the ferritin. Okay. Now, deficiency of iron. Iron deficiency usually results in microcytic hypochromic anemia. Okay. And it is the most common form of anemia in the US, resulting, and it is as a result of decreased hemoglobin synthesis, and consequently, the red blood cell size will decrease. How is it treated? Administer iron. Here you can see normal red blood cells. You can see how they are large. Their mean corpuscular volume is, can appreciate how large they are. But here you can see that these cells are small, so microcytic, then they are pale because they don't have enough hemoglobin, hypochromic. Okay, so this is normal. This is a patient who is suffering from microcytic hypochromic anemia. Now let's look at iron overload or excess. Now, iron overload can occur during accidental ingestion of iron, where, and please note this, that acute iron poisoning is the most common cause of poisoning deaths in children below the age of 6. Okay. Now, the upper intake limit of iron is 40 milligrams a day for a child and 45 milligrams a day for an adult. Now, any level beyond that is toxic. Okay. And it is treated by use of an iron chelator. Now, overload can also occur with people with genetic defects. For example, people with hereditary hemochromatosis, okay, HH, which is an autosomal recessive disorder of iron overload, which is primarily found in those people who have a northern European ancestry. No countries in the northern European, such as Denmark, I think Iceland, such countries. That is where you find this autosomal disorder, autosomal recessive disorder prevalent. Okay, and it is usually caused by mutation of the HFE gene, the high iron gene. Okay, how does it present? It presents with hyperpigmentation, also hyperglycemia. So it is known as the impulse bronze diabetes, bronze because of those hyperpigmentation, and the diabetes because of hyperglycemia. Also, there will be damage of the liver because it's the major storage site for iron. The pancreas will be destroyed, the heart may be destroyed, and in hereditary hemochromatosis, the serum iron and the transferrin saturation are usually elevated. So you can find instead of 33% of transferrin saturation, find like it is 50%. Okay, that you will know that there is some elevated levels of iron in the body. Now, treatment is by phlebotomy. You can remove blood, and also so as to reduce the concentration, you can also use iron chelators. Now, iron overload is seen in also can be seen when you have mutation of proteins of iron metabolism. How? Let's say one has a mutation of hepcidin, okay, resulting to low levels of hepcidin, inadequate hepcidin. When there is low levels of hepcidin, the body assumes that you don't have iron in the body, but in this sense, maybe you have. So ferroportin will be transporting more iron into the body, so that will lead to increased, there will be iron overload, increased serum iron levels. Okay, and this can result to hemosiderosis, and that is there will be deposition of hemosiderin. Hemosiderin are aggregations of ferritin. When ferritin aggregates, it forms hemosiderin, which is insoluble, which is an insoluble storage form of iron. And remember, it can occur in the liver, and that is why it can lead to liver damage. Okay, and I think guys have come to the end of that session. And thank you for your attention, and please subscribe for more anatomy and biochemistry content. Thank you.